So I'm James Vornov. So I'm an MD, PhD neurologist, and I had a pretty long academic career. I was on the faculty at Johns Hopkins for about 10 years. And during that time as a neurologist and having a lab, I moved further and further over towards having further and further to really wanting to do therapies. And it became frustrating as an academic, and now this was quite some time ago. It's actually 25 years ago now. We didn't really have the resources, so I realized that I'd be better off in a company that had the resources, medicinal chemistry and toxicology and all of that, to actually create therapies. But it's interesting, so when you're an MD and you move over into industry from academia, really where they want you is in doing clinical research, running trials with the patients. But for me, it's actually been a great career choice because I stay close to the science, and especially now I work mostly with biotechs and early stage development. I stay close to the science and then also work on this very hard problem of actually translating in the clinic all of these potential therapies into actual treatments for patients. So, well, a couple of years ago when I joined the faculty, I talked to Barb and Mike about, what to talk about. I realized I had an audience of these future drug discovery scientists. And so of all my experience over the years of working with biotechs and seeing products and seeing them succeed and fail, it's like, what would I really want to tell this group? And so what I really want to tell you is summarized really at the very beginning, which is that as you're actually doing your science and finding molecules and treatments, you're already in the process of drug development. It started already. And I sometimes call it the DNA of the product in the end is being laid down. It's the fetal form of the drug that will eventually be on the market. And so it's sort of path dependency. Whatever you do at that point, it really casts the die for what can happen, in the future. So what animal model you pick. So if you work in a lab working on ALS and you have a mechanism of cell death, and so you only do ALS, you're creating an ALS drug, but the truth is that it may have broader applications. It might actually work better in a different animal model. So thinking as a drug developer, you're thinking about the compound and the treatment and the potential product, not the science of a particular disease and how this compound informs the disease. Route of administration, right? If you have an oral drug versus an IV drug, an oral drug can be used chronically, but it's slower acting. IV drugs act fast, but they're generally hospital-based clinical drugs. Biomarker development is very, very important to us clinically because what happens is we'll get a drug and start development and say, boy, it would be great if we had some blood biomarkers or nowadays CSF biomarkers or imaging biomarkers to tell whether or not the drug's working. And well, if the groups from the very beginning had this in mind, is how do we tell that this is working without sacrificing the animal and looking at markers in the brain? If they thought about, can we image? What are the markers? Can we develop other ligands or other ligands useful? Then we're set to go in the clinic to know whether something is likely to be useful during clinical development. And so often we can't use that because there's not enough of a knowledge database to use them in the clinic. So I'm like the guy, we used to call it throwing it over the wall. I'm the guy who's going to get that package of all the knowledge about the molecule that we're not going to put into people. And so what I want is actually the best thing is open communication. This is actually when I'm hang around Barb's group, is this communication with talking to the scientists and the chemists about my clinical perspective in neurologic disease and psychiatric disease and pain to figure out what are the characteristics as you're selecting a lead. What is it going to look like and what's going to be the utility and what are the challenges that we're going to find years down the road so that you can anticipate them. So if there's one message that I have for everyone as you leave, and I think a lot of our interactions at the roundtables, I'm always talking about this, you have to think about where you're going. And so what I'm going to talk about now are some tools and basically heuristics about thinking at an early stage about where you're going and how to become also more knowledgeable about where you're going. So you'll hear us talk all the time about drug labels. So the label is not the sticker that goes on the box or the bottle. It's this, which, they actually call it patient package insert, right, very often. But it's the product of all of the development, all of the data, submission of the new drug application, the NDA, or the European filing for approval. What you actually end up negotiating with the regulators is what's on this piece of paper, which is all the constricts of then when the company goes out to market it and present it to physicians and patients for use, they're only allowed to talk about what's here. So physicians use things off-label all the time, but companies get in a lot of trouble, billions of dollars of trouble, when they cross that line and start trying to promote a drug for things that are not specifically in the label. And also the dosage and the use and the basic information is all here. So this is actually, nitroglycerin, about the oldest kind of drug you can imagine. They've changed them now. They're a little bit easier to read, but the basic information is always there. And if you see the one thing that's what we can focus on the most, because this is like how to use the drug and how does it come, et cetera, but it's the indication. That's what we're looking at from the very beginning, is what does the label say this drug is for, what patient population, because that's who's going to be able to. And I should also mention that over years, payers have become a big participant in what you can use a drug for. So it first goes to the FDA, but then the payers will say, well, that's not in the label, we're not going to pay for it. Or they'll put other strictures about dose and what was actually in the label, because they'll use publications as well to make their decisions, but the label stands. So labels vary broadly. So these are two neurologic drugs that I always think are very interesting labels. So Topamax started out in epilepsy, and you can see it has very, very detailed, right? So it's for initial monotherapy in patients who are older than 2, generalized tonic-clonic seizures, but then pediatric and adults 2 to 16 with partial, and then Lennox-Gastaut greater than 2. So you can see that they never tested it on patients less than 2 years old. So in the label, there's no information. There'll be a warning or some caution that it's not been used. Now, physician can use it if you're 1 year and 11 months old. But the label in the trials, it was only used for 2-year-olds. And then later on, it was discovered utility in migraine, and so it has a separate prophylaxis of migraine 12 years and older. Aricept has this thing of a dream label, right? It's the broadest label you can imagine for Alzheimer's disease. It's for the treatment of dementia of the Alzheimer's type, mild, moderate, severe. Now, mild, at first it actually only had a label for moderate and severe, and then they actually updated the label when they did studies in mild patients to include that. So that's what you're aiming for. The end in mind is an indication that says this is for the treatment of this population's disease. And so years ago, the FDA and industry came up with what is a very, very useful tool to help you from the very beginning get to that end, which is a target product profile. And so this is an example of what one might look like for a Parkinson's drug. So you can see up top is the indication. And so you have a drug, and this happens all the time in neurodegenerative disease, right, is that you put it into these, MPTP models, 6-hydroxydopamine models, and there's more movement and better function over the course of a brief period of time. So that would be like symptomatic treatment. You treat a patient and they get somewhat better on the scales. But of course, what you really want would be disease modification. You take it for years and you actually prevent the disease. Much more valuable therapy than yet another symptomatic therapy for Parkinson's disease. Now the population, you may have a mechanism that is focused on some particular gene or mechanism that's suggested by a gene saying it's a risk factor. So you could do a trials where you only go to the patients with that gene, and you may have in the label, probably suspect you'd have in the label, right? It's only for patients who have identified this genetic abnormality. But of course, what you really want is everyone with Parkinson's disease, even pre-symptomatic patients who will get Parkinson's disease to prevent them. So, and, oral, formulation, think about it at the very beginning because it may be easier for one lead versus another. In Parkinson's disease, oral disintegrating tablets are, because of swallowing problems and oral problems in the patients, they're valued. So if it's very potent and small and would dissolve and be usable that way, it's going to be a more useful drug down the road. And then we'll talk about regimen. And then these are the kind of endpoints that you're thinking about. How are you going to measure the effect down the road, which is all disease specific. So, how do you show efficacy? It's a constant moving target. It changes with medicine. Very simple that, in 62, it was this adequate and well-controlled studies. And so the fact that the law had plural really made everyone believe that you had to have 2 studies and that became the tradition. And then PDUFA, other laws, it really is possible very often to have 1 clinical study and good supportive data that bucks it up so that your statistically significant result is very likely to be true. And this, it's risk benefit. And so how much risk you have versus what benefit you can show is in the end what they approve drugs on. And so if you have a risky drug that has a good benefit, but it is not going to affect patients' lives very much, it may not actually get approved. And of course, we've seen in recent years approval of drugs that have minimal or doubtful benefit and with it to be further characterized in the clinic over years. So the other tool that we have from the beginning, and this is only at the IND stage, is that we start collecting all the data that we know about the drug in this document known as an investigator's brochure. So when you start IND-enabling studies, that's what you'll be collecting for. And it's really the draft label, so what's in there in terms of safety and pharmacology is all what's going to be in the label. So it's useful to think about that. And some of the information from your initial IB will actually be in the label, but it supports the investigators and folks like me who are running trials to know what is known about the drug, because I don't go into the data of the companies to look at what their preclinical data look like. They may have publications, they may not. It's all just here. And so this is should be a concise, non-salesy, good argument for why this drug may be useful in the population. So there are a lot of resources. These FDA guidances, I showed one, they're not updated often enough, and they vary from being really well written and well thought out to being kind of, here's what we think, and some of them are really quite old, and it's a wonder that they don't just take them off, but they don't have anything to replace them with. And they generally don't lead the science. They're generally, after they've done a bunch of approvals, they'll then, like someday they'll probably do a guidance for approval of Alzheimer's, disease-modifying Alzheimer's drugs, since they've been through this now a few times to kind of put it down in writing, because they're giving guidance to companies all the time. Before you go into the clinic, generally you'll have a pre-IND meeting with the FDA, and you'll say, what do you want to do, and here's our data so far, and have a conversation about what's going to happen next. Well, that's guidance from the FDA. It's just not written down, and no one other than you and the FDA have access to it. So very, very valuable is looking at all of these labels. I do that all the time when we get a new program and I want to think about what kind of endpoints are going to be useful in the trial. I go and look at other compounds that have been approved either in that indication, or what's been very, very useful to me over the years is indications that are the same or have some resemblance. In one of the, we got thrown a trick for the FDA years ago where they wanted efficacy for a drug that was clearly efficacious. And where we ended up designing the trial was from the antibiotic literature, antibiotic labels approval, where they use dose dependency to show that a drug works, rather than giving patients nothing. And that worked out well. So we talked about it a little bit yesterday, actually. So online, and they're all online now, thankfully. We used to have to order them, or there were services actually that had them. But they're all online, these summary basis of approval. The problem is they're only drugs that get approved become public. So it's for the public good, right? You know why the FDA approved this drug, and advocacy groups and physicians and anyone who wants to can go and look at. It's got the summaries by the medical reviewer and the pharm tox reviewer about what the data showed and what they thought about it. There's also, when there's advisory committees, all those slides get put online as well. And that's also gold in terms of understanding what's going on in drug development. The problem that we have, of course, is that all the drugs that don't get approved, it's all buried and you have no access to it at all. Academics who participate in drug development, so I'll use Mike Rogaski here as a good example. I mean, he's had a very long, successful academic career, a lab, basic science work, but also he's founded companies or worked with companies over the years. So if the areas that he's worked in, you're entering into those, you just call Mike, send him an email, and you can find out who these people are pretty easily. And they do this as part of their career, and they are very, very approachable in general, because they're always interested in what's the next thing, and they want to get in on it, they want to consult on it. And so they are very, very valuable resources. So I personally kind of have a network of folks I've developed over the years who I know are reliable and honest, that I can ask questions, and I'll just send them questions that are blinded to what the compound is or the company, and say, what do you think of this? We're trying to do that, and they're always happy. We're always happy to talk about things like that, because it's our lives, right? It's our scientific and careers. And then, consultants, CROs. So actually, I didn't mention, so I worked in biotech. The company I work for now is a contract research organization. We are a service company, and what we do is work 95% with biotechs, and we run their clinical trials. And so I'm the physician, the medical monitor on a lot of these trials. So companies come to us and say, here's what we're thinking about doing. How much will it cost? Is it practical? How long is it going to take? And so we have the experience to tell you that. And we do that actually for companies for free as part of the process leading up to them actually engaging us, because we want to demonstrate to them that we understand the space and we know how to do the trial. So that's actually a big part of my job, is business development, talking to biotechs about what they're thinking about doing. But again, for me, it's all information. I get to see what people are doing, what are they thinking, and what's being funded and what's up and coming, which helps our company position ourselves for the next. So the next part of this talk is I'm going to give you my personal heuristic for how I look at clinical development. I was taught this years ago by a medical writer, and I use it virtually every day. I've used it twice today already in answering emails and planning some stuff about clinical development. So clinical development, so you're going to try to understand where your drug is going to go, right? Even in this early stage that you're at. And so clinical development, doing all of this, there's lots of moving parts. And yes, it's really complicated, it's really challenging, and there are huge numbers of problems. But in the end, it's really very, very simple, and it boils down to just three things, which I call the ABCs of clinical trials. And that is, you take a bunch of people, you get a population together, right, that you're going to put into a trial, and then you do something to them. Usually you do placebo or active, or one active or another active, right? But you do something to them, and then you measure what you did to them. And usually what you did to them, you have to measure them before and then measure them after, but actually not all the time. In stroke trials, we only measure outcome. We qualify patients to come in because they have some injury that's probably going to have an outcome, but we don't know what their outcome is going to be, and then we measure it afterwards. In Parkinson's disease, it's what's their UPDRS score before they come in, or what's someone's seizure frequency before they come in, and what is it on the drug, and that's the outcome. But it's simple as that. And so as you work on your projects and talk to people, just keep this in mind. Who is this drug going to treat? How are we going to administer it? How are we going to give it? And then what are we going to measure? So the population. So, Now, this is the traditional way of looking at of populations, right? You can treat a syndrome. So much of psychiatry, right, is treating syndrome, psychosis, low mood depression, right? You can treat, the substrate, the anatomic, biochemical, pathology. And of course, sometimes in disease states, we know that one's operative and another one, right? If someone has diarrhea, you can have a diarrhea drug, or you can treat their IBD, or you can treat their their infectious disease, right? Or some particular cellular vulnerability. And you can see that when you start getting to substrate, it can go across multiple diseases. Then, of course, pathogenesis, especially like genetic risk factors, environmental risk factors. And then what, very often we're trying to find, the silver bullet to cure disease. And so that's these final common pathways. So, amyloid in Alzheimer's disease, and prevent the pathology. But also, final common pathways is also mitigating symptoms. So I don't know how all these patients end up with their substantia nigra degenerated, but they all look the same in terms of Parkinson's disease. They have different genetic mutations, probably environmental disposal, but I can't have access to that most of the time. So, but they all benefit in the common final pathway from restoring dopaminergic function. So in drug development, we're in science, we're often in this area, and in drug development, we're often trying to find broadly useful drugs that work on large, because all of these things then converge on certain, and I should just say that those are often the most difficult, right? They don't really work very well because it's very hard to intervene in a lot of these biological processes. So just to quickly talk about where we are now, and or I mentioned it this morning, precision medicine, right? We are there now, and so much of what we do now is using populations that are defined by genetics. Now, and neurologic diseases is very, very rich in this. Psychiatric diseases are also, we just don't understand the genetics of psychiatric diseases very well, but we certainly know it's there because we know that all these things in the brain run in families, like much of medicine, right? Diseases run in families, and there are twin studies. You could see that these diseases were heritable. And then, over time, we've gotten single gene mutations, Huntington's disease. But then there's lots and lots of other things where it's genetic, or even you understand what a gene is in a disease, but it doesn't explain all of the disease. Or you find the gene and it doesn't explain how it's linked to the disease. So it's just like a little list of some examples of that. I don't want to take the time to go through it, but I say like, Wilson's disease, is a copper transport gene defect, right? And so it's a copper disease, and you chelate the copper, and that's generally how we treat it. Now what you try to do is go fix the gene. So that's how things have moved from common final pathway of copper metabolism to, to the gene itself. Huntington's disease, a autosomal dominant gene. One of the first triumphs of finding the gene of a neurodegenerative disease, and yet I personally think that today, all that time later, we still don't understand why what CAG repeats are actually doing, and how it affects the brain and causes the injury. And so we're actually now in a phase of treating Huntington's disease where we're going right after that gene, and we're not worrying about all the mechanisms of damage and the pathways to get there. And some of these diseases are getting sliced into, right? There are many. Autism is a broad disease now. There are lots of genetic abnormalities that we always classed into autism. We knew that patients look kind of different, but now we're starting to understand that. So I think we're at the beginning of the process here, but it is definitely where drug development is going. And because of success in rare disease, there is very little hesitation to go after specific gene defects in small populations. So, Alzheimer's disease, that's a place where we've learned a tremendous amount about who has the disease. We initially, all of our trials early on, were treating patients who didn't actually all have amyloid disease. But it's still very complex, right, in terms of all of these stages in the disease. And so, we're mostly at this final common pathway, but see companies going after, ApoE for treatment to actually try to intervene in that pathway, since it seems to be pretty robust, and understand how it actually works. So you'd only do those trials in ApoE for homozygotes, for example. So it's not going to be a broad treatment for Alzheimer's disease. The ALS frontotemporal dementia is really kind of mind-blowing as we started understanding it, because actually it was known that there were these sort of bridge patients who had symptoms of both, but that there was a single underlying gene in patients who had completely different looking neurologic diseases. We don't understand why some patients get one and some patients get the other, but, and now there's other ALS, FTD. So clearly that's telling us something about the biological mechanism of how the disease occurs. But for us as drug developers, these are expected to be levers into how we're actually going to treat the disease. And so, so we have a lot of genes, the same syndromes, and actually, we've run a trial, and so this has come out of oncology originally, right, where we know that there are genes that drive different kinds of cancers. It's like, why did one person get a cancer in their lung, another in their prostate, another in their kidney? Well, the trials are run now is that you identify the gene, and then you put patients in a basket trial, it's called, who all share the same genetic defect for the same drug, right? Because it's expected to work well across those. And then often it gets narrowed down and you do a more traditional this is a renal cancer trial, this is a lung cancer trial, but to start as an exploratory way of doing it. So this is a perfect example of where you can put patients together who have frontotemporal dementia and ALS driven by the disease. It's more patients to look at, and maybe it'll be a better readout in one versus the other because ALS is faster or FTD has more cognitive, perhaps easier to change than restoring motor neuron function. We don't really know that, and so in the early stages, that's one of the places where things are now going. And so, Parkinson's disease as well is it's one of those areas where there are so many genes, and some have big impacts, some have small impacts. There's another problem is that, are these risk factors? Are they driving it? Well, in Huntington's disease, everyone who has, it's autosomal dominant, everyone who has it essentially will eventually get the disease, and right, we know about anticipation, so there's like different doses there. But for a lot of these others, and so one of the questions I always ask folks when they come up with these new treatments, I always ask them, well, how prevalent is in the general population? Because a lot of these genes are actually relatively common. So you then start looking in the disease population, you say, oh, these patients have this gene that is a marker for those, but there's another 10 times as many people in the general population who never get the disease. So the question is, how strong a driver is that of the actual disease? So if you change it, maybe it's just like a risk factor, like smoking. Sure, if you stop smoking, you'll lower your chances of lung cancer, but it doesn't really help you once the lung cancer is there from your smoking. So now we talk about precision medicine, but I think that one of the big things just to take away is that now you can think a little bit more about your targeted therapy, and are you going after, again, just kind of the syndrome, these behavior signs, and or are you going after a mechanism that really demands this kind of genetic diagnosis, other genetic factors, environmental factors, or a particular mechanism going on in the patients. That's one of the things that we're now thinking a lot about, is that based on risk factor or these gene profiles, can you identify a patient with Parkinson's disease, maybe who's driven more by mitochondrial dysfunction versus one that's driven by cytoskeletal, lysosomal problems, because the genes are falling into these buckets. And so if you have something that's mechanistic at one of those levels, it may be better to pick a patient population that you believe to be more likely to be evidencing that mechanism than just the overall population, or at least early on characterize the patient population. So when you analyze the data, you can get some idea about whether or not it's working better in one group or the other, which is generally the approach I tend to like early on, because you get more safety information, you get more understanding. And sometimes if you only go after one population, you'll never know until way later, or possibly never, that it would have worked more broadly because of the mechanism of action of the drug. So actually, Greg gave a great introduction to talk about animal models, so I don't actually have to say anything. But it really is that we will get drugs in the clinic, and the question is, what do we actually know from the animal models? And so very often I will tell companies that, you don't have a model of Alzheimer's disease, you have a model of amyloid overexpression in rodents. And so if you overexpress it, and then I go after that target because I overexpressed it and it causes animals to have problems, yes, I've demonstrated that that mechanism will rescue my animal overexpressors. But it's not the disease in humans. The disease in humans, right, has genetic, environmental components and isn't manifest until someone has lived 65, 80 years of their life. And that is not what's going on in animals. And so, there's a lot of kind of disappointment, and the animal models aren't validated because they don't translate. And when people talk about that, it drives me nuts, because it's really that they're not thinking deeply enough about what they're actually doing with their animal models. So, I always say the best thing is that when we have an animal model with some biological mechanism that we have working in the animal model, and we can actually measure and see that mechanism in action in the clinical population, then I can very directly take the drug, and sometimes what I love is in the first clinical trial, demonstrate that my drug that worked in animals does exactly the same thing in people, and I know what the dose that I need for it, and I know the duration of action on that mechanism. Then it's a matter of translating it into clinical benefit, which I'll talk about. So how will it be used? Again, the first clinical trial I always tell companies is their first marketing study. And so when you go into the clinic, it has to be for real patients that exist and can be identified. It has to be an unmet medical need, and it has to fit into the standard of care. And so it's really just to point out that a lot of times talk to people early on, they have these dreams about how the therapy is going to work, and they haven't thought at all about how it's going to be administered, how it's going to get there. And we have gotten much more inventive and much more direct now about how we do things with intrathecal and intracerebral injection. So the world is a lot more open now than it was even 5 years ago because of some pioneers in the field. But you really need to think about this molecule that you're now putting into animals, is how is it going to be fit into a clinical trial and into standard of care. And of course, by the time you get to the clinic, standard of care is likely to have changed. But you want to try to anticipate some of that. So again, it's sort of metabolism also, interaction with other mechanisms, how these are treated. And again, this is stuff that, our clinical colleagues, folks who work in the trial and who are academic and understand, and folks like me would love to talk about this, because we can give you some insight into what's the current treatment, how would your mechanism, et cetera, fit in. So all I'm saying is have those discussions early on and some foresight, because, again, decisions that you make at this early stage are going to have consequences for the folks who have to actually deal with this in the clinic some years later. It's remarkable how little we think about route and frequency. We have all these drugs that go in animals that are dosed 5 times a week and not on weekends for chronic treatment. And then they go into the clinic and say, well, it has a short half-life. We have to give it 3 times a day, 7 days a week, right? And it was never given that way in animals. And you go like, well, why not give it the way it was given in animals? But it's very hard to explore that in people. And so you want to explore it in the animals. How frequently does it have to actually be given? How long does it last? And again, the more you understand mechanism and have ways of measuring mechanism, and think about, can I measure that in a person, in their blood, in their CSF, through imaging, the better off everyone's going to be through the whole. And again, one of the things that we don't talk about as much anymore is you can quit early. If you have something that worked in animals, you get it into people, you expect to see it to work in people and do something, and it doesn't do that thing, well, you probably ought to stop at that point, because your reason why you're developing it is because it did it in animals, now it's not doing it in people. Maybe it's not getting into the brain, maybe the dose isn't, maybe the side effects are too great to get to the right level, but it's really very, very useful in early development. But once you're in early development, it's too late to think about these things. And then outcome measurements. And I think I've actually talked a good bit about all of this, already, but, it's sort of how are you going to deliver the treatment, confirm that it actually got there? And, we have imaging techniques and, receptor occupancy, but you have to have generally a PET ligand already in order to measure it. We measure CSF concentration, but it's good to know what the CSF concentrations are in animals compared to the measured brain concentrations. So when you get to people, you only get their CSF concentration. So if they don't match in the animals, then you expect they're not going to match in people. So when it's not in the CSF, you won't be surprised. You won't try to measure it at all. But if it's more in CSF than in brain, because it's somehow secreted or accumulates there, then it's good to know that as well. So, we talk a lot about biomarkers, and I just talk about, biologically, how do you know that it's working? And because people get all confused about some of these things, right? A biomarker broadly is just any biological measure. It can be in blood, it can be in imaging, it can be anything you like that just shows that you have a response, right? It's a marker of biological effect in essence. And anything like that we can do in people is useful, but you have to develop it in the animals. And actually, your compound, your program, is more likely to go forward when there's that kind of ability. So it's really gaining knowledge over time in the animals and then in people. And you're not going to use this biomarker for getting the drug approved. It's not going to be the label. So I just want to go through a couple of examples. So this is one of my favorite recent examples, although in the end it's kind of unfortunate, is this ASO that Ionis put into the clinic for Huntington's disease, eventually was licensed by Roche. And so it's really fascinating. The first study, this is a pharmacokinetics of the ASO in CSF, right? So here's the dose, and here's how much is there. And this looks really bad to someone in early development, right? It looks like you get the lowest dose, you get a certain amount, and then it's just like flat after that. So it's nonlinear. There's a cap on it. It's like, and actually in a lot of drug development, we would just dose here because there's no reason to dose higher, right? But because there was a readout of the effect, it turns out that if you actually look at suppression of mutant Huntingtin in the CSF, there's a very nice dose response. It's actually probably the highest two doses, not much different, but these doses where the kinetics, pharmacokinetics all look the same, there's actually a dose response on the biological readout. And if there hadn't been a biological readout, this would have been done completely blind. And so it went on to phase 3 trials, and this really works biologically to lower mutant Huntingtin, right? This is out to 421 days. And then in the phase 3 trial, however, the problem is that it didn't actually improve patients lowering it. In fact, it's not statistically, and this is all official Roche documentation from their annual report stuff on the internet. So it's all publicly available. It's not been published as far as I know to date, although I haven't checked lately. But there's actually a reverse dose response. The highest dose actually tended to have a worse outcome than the untreated patients. Patients decline over time anyway over these, it's over a year, it's 52 weeks, and it's a little over a year. They do decline. You can measure the decline. The studies were really well conducted in terms of measuring decline. They were big enough, but the treatment. So suppressing a non-specific ASO suppressing Huntingtin doesn't improve patients was the conclusion. Now, there are some subgroup analyses, and they were going to go after those, but I haven't heard much news about that afterwards. one of the other things for safety was that actually there's, again, a dose-dependent response, with the highest dose being the worst in terms of increased ventricular volume, so volumetric studies in the brain. So again, this drug is biologically active. The ASO is biologically active, decreasing, but it didn't work, and actually there's a safety. And then it did actually reverse. They followed the patients afterwards, and this ventricular increase actually reversed after the drug stopped being given. So now this is a graph from a label for amyloid depletion in Alzheimer's disease, where again, very nice dose response of the antibody over a year, depleting amyloid from the brain. So again, biologically, the amyloid depletion antibodies, took a little while, but they really, really work. And you can figure out the doses of them and get to safe doses that deplete amyloid. And this is the graph again from the label of one out of the studies showing clinical benefit, and that's really the basis that it was given approval. So this is Aduhelm, which you can look at this later, but this is the indication and uses, has this long, it's approved under accelerated approval pending further clinical. So it was, but it was approved just for the treatment, indicated for the treatment of Alzheimer's disease. Should be started with mild cognitive impairment. Nothing's known about starting it later. So that's actually how Biogen and the FDA negotiated for this label. It doesn't say anything about how you have to have an amyloid positive PET, although Bart tells me that that's actually how it's being viewed by insurers, because the trials were all done with patients had to have positive amyloid PET. That's who they want to give it to, not just anyone with mild cognitive impairment. So let me just finish off at the end, with how to pull all this together. So the chief business officer of the last company I was with liked to talk about this. It's sell the dream. So you're early on. No one knows if it's going to work. It's research, it's science. But one of the advantages of looking down the road about what your product is going to look like, who it's going to treat, how you're going to show that it treats them, is that when you talk to funders, investors, grantors, anybody, you have a clear idea of where you're going. And it's very much about the story that you want to tell, right? About now they'll have all of your IND, the IB, and the protocol, but really what you want to do is have some vision of what the step-by-step path is to that ultimate label. And that's why these tools and heuristics, I think, are very useful early on to understand and understand the value inflection points, because one of the things that I love about a drug where the first time you put it in people, you can show it does in people what it did to animals, is that although the venture capitalists will kick and scream that they want clinical outcome and P less than 0.05 on that early on, they get very, very excited because they know that the probability that at the end it's going to work are very high. Now, Roche looked at the Ionis product and that early suppression of Huntington and said, this is going to work for sure, right? Well, they had to do these very long trials to know whether or not the biological effect, let me just emphasize, they are two different things, right? Your drug working on mechanism in animals and even working on mechanism in people is different than clinical benefit. And what we clinically end up needing is clinical benefit, but that's something you can only do when you're actually in the disease, itself. And so your confidence and understanding is what becomes infectious for the people who are going to give you money and help you get there. Whether it's selling it to a company or forming your own company or doing it all academically. So I would say every product needs a champion. That's how so much of this ever got into the clinic. I think actually probably you saw that last night, that there's a lot of serendipity and the right people, and those people see that it's going to work and they overcome all of the challenges and difficulties. And believe me, that's what drug development is. It's difficulties and challenges day after day. And so you want to become as expert as you can. in the clinical development pathway. You don't have to be a clinician, an expert clinician in the diseases. You want to understand the biology and how it fits. So, read some of the guidance, look at some of the guidelines, talk to experts, go to meetings, listen to the talks about other drug developments or other developments of treatment so that you can map out how you're going to get there. And so I've always was taught in drug development from the early beginning, when I left academia and was have a sense of urgency, right? And I think the best way of putting why have a sense of urgency is not only the payoff and the money, but also that the patients are waiting for these treatments and that it's long and difficult. And so the faster we move, the more lives we'll be able to save and the better lives patients will have. So thank you.
Okay, questions for James?
Thanks. That was great. So I just have a practical question about when you're drafting a TPP, right, the target product profile. Is there a preference for whether you state that your goal for your product is to alleviate some negative symptoms or to improve some desirable outcome?
Yeah. Well, it's going to depend on that's why you have to think about what your clinical trial, that's why it's so funny, right? You're starting off, but you're going to think about what the clinical trial, so let me use an example that comes up over and it's negative symptoms in schizophrenia, right? Are you going to try to alleviate negative symptoms or are you going to try to improve cognition? And so the problem is that improving cognition isn't a clinical benefit as viewed because improving cognition is generally doing better on some kind of a cognitive test and that's not, and so when we actually, so there's a scale called the PANSS, right? And so improving negative symptoms is improving the PANSS, but if you actually want to improve cognition or executive function, right now how we're postulating all these things, you have to figure out where you're going to go. And there are pathways. Unfortunately, there's something called the Matrix, which is a group of cognitive tests that's got put into everyone's minds early on before we really understood things very well, and it's not very good as a clinical endpoint. So, but we have clinical endpoints invented all the time. Actually, you think about the whole SMA story. You're treating infants, you're treating young children. How are you going to show those kind of improvements in a scale? And did they even expect that they were going to have children who were, flaccid at birth end up being able to over that kind of time? So how do you measure that? So a lot of we don't understand how to measure. So sometimes the measurement comes along with drug development.
So if I understand what you're saying, you kind of need, we academics kind of need to understand the clinical language of like what does a patient improvement look like so that we use precise terminology. In this.
Right, yeah. Or understand what people are thinking about. So like in the schizophrenia example, how are people thinking about negative symptoms? How are people thinking about cognition? How would you improve the life of someone with chronic schizophrenia?
Any other questions for James?
Because I know you target Huntington, which is in my heart. I was always looking for a target in Huntington disease. But the question is, this is a rare disease, and so the company, a lot of companies, they may not be interested because it's a small population, so how you get away from that? I mean,
So actually, I took some of that out, because one of the real big changes that occurred, years ago now, was biotechs realized that they could go after rare diseases and have drugs that had big impact on the disease. And when they had big impact, they could charge a lot of money for it. And the trials were short, or small at least, because if you have a drug that's only going to treat 10,000 people around the world, your trial then only had 50 patients in it. But if you're going to do a trial, and sometimes they didn't have placebo groups, they were based off of natural history. And so you could do a 50-patient trial in a rare disease, get your drug on the market, and charge $100,000 a year for it. And so that suddenly changed the math. And so we exited years ago. Big Pharma was interested in blockbusters, right? Drugs that treated everyone and had no side effects. And Big Pharma more or less collapsed when the stream of blockbusters ran out. And then biotech took, and now, Biogen and all these companies, they're interested in Huntington's disease, or rare, which is relatively rare disease. And so, everything's changed. The challenge, though, is that if you're going to go after one of these small populations with a really targeted therapy, the expectation is that it's going to have a very big effect because you're actually going after the target of that therapy. And so, like you can see, these Huntington's trials, in the end, ended up being pretty big and pretty long, but tremendous support. And actually, one of the other things, tremendous support from the community. So if you're going after a rare disease and there's good patient, ask anyone who works in these kind of rare diseases, patients and their families come to you as an expert saying, we want treatments. And so they end up networking with all the other families who have the same genetic defect in autism or metabolic disease. And so doing the trials actually turns out to be, in a lot of ways, easier than they are to find patients with more common diseases, but who are more distributed. And you got to entice them to come into a clinic and be in a trial for a year. These patients are highly motivated and very well educated, and you know what's going on through patient advocacy groups often.
Just a quick, over here, sorry.
Yeah.
Just a quick comment on the Huntington's. I mean, I think, I believe you said it, but just want to make sure everybody heard that clearly this drug was not able to distinguish between the wild type Huntington and the mutant. And the hypothesis was that knocking down the wild type was not going to have an effect, but clearly there's some additional complexities here. So the drug did what it was supposed to do.
Beautifully.
But unfortunately, it did not lead to the proper outcome. So, and the other, I guess, quick comment is regarding what you showed on the CSF. In the level of the ASO. Do you know when that was assayed? How far after?
Pretty long after, because that was one of the issues, and it comes out of the nusinersen as well. Is that no one really thought about ASO pharmacokinetics at the beginning, right? We were rushing to figure it out, and so. Now, actually, in most of the programs, we're really having to look more closely at when you administer it over time, what actually happens. But when this was it was after it was the first time going after this with
I think the complexity is the sampling, the PK sampling. What's really known about all the ASOs, and we saw that, and as well as some of the other Ionis drugs, obviously, is that the drug gets into the CSF, but say 90% of it rapidly goes out, and it's actually in the systemic compartment essentially. And your PK is going to go up, down, within probably a couple hours, and then you have this long tail. So what you're measuring in the CSF
and actually,
is the long tail, basically. So, it gets harder to look at that difference.
And actually the point I'm always making to folks is that if they hadn't had a biological readout, how in the world would they ever have known, right, at that point, what to dose? And so we're very often flying blind. And so the fact that there was a readout and they knew how much they had to suppress in animals in order to get was what guided this, and what I think is a beautiful scientific story. Unfortunately, it didn't work out right because, again, and when I'll never forget when we first, looked at this and said, what in the world is going to happen when you suppress all of Huntington in these patients, right? I mean, are they depending for every other cell in their body on the non-mutant Huntington? Well, when you do it to animals who are like overexpressing, well, it doesn't hurt them. Well, is that the same thing as taking one of these patients and having their whole lives lived with this mutant Huntington? And it's remarkable that really, but let me also notice that I think one of the other areas we are really at a problem in the whole area is that when you talk about, well, it distributes out, right? We're learning this in animals, right? But in people, we have no way of looking at it. So when we first start seeing the CSF data, like, is that just superficial spinal cord? Is it, just the convexity? Is this actually changing expression in the striatum? There's no way to know in people without, autopsy. So we have to make, I think in the end, I believe that it was, because we see probably changes in the disease and we see these other effects. So I think it probably does. It's very, very powerful evidence that ASOs for this kind of CNS disease are a reasonable strategy going forward. But right, again, between the mechanism and the clinical benefit, it's a hypothesis, and this is clinical research, clinically testing that hypothesis in the actual human disease. And you can never forget that that's the ultimate test. So you have to, as always, get into the clinic and test it in patients. There's
yeah, the hypothesis was clearly tested, and unfortunately it was negative.
It did, and it was negative, right? And that's science, right?
So just follow up with the rare disease now. If we move to psychiatry more for addiction, like alcohol addiction, or how we can change the culture, because so far for addiction to alcohol, we're just relying on academia. Pharma, they're so that's the challenge. I mean, for we have only how many drugs we have in alcohol addiction? We have 3, maybe to 6 if we include Europe. But it seems like the pharma, they are not really interested in moving forward drugs. So I'm not sure how we can change.
So I think the innovation, thankfully now in CNS now, is in biotech. And so it's the people coming into companies and folks. And so I look through I think I'm actually the only MD. Mike is on faculty, but I think I'm the only other MD faculty member. But we have a bunch of MD PhDs who are in the taking the course, and it's always been true over the years. And so, a lot of those folks and the folks who aren't here, who are more on the clinical side in doing the research, they are, when I moved over to the industry, it wasn't so common. It's actually a good bit more common now. Establishing a biotech, working at a biotech, and becoming that therapeutic visionary of how we're going to change therapy. Because, again, I'm going to go back to Ionis over and over again. I talk about this all the time because what an incredibly brave thing it was for them to do as well. They said, let's forget about the animal models and figuring out whether or not let's do the clinical experiment and see whether or not it actually works. Now, they had nusinersen as probably going to work, but in Huntington's, would it actually work? And so that's a lot. Now, in psychiatry and drug dependence, I think, again, there's a lot of creative thinking that has gone into where, like just binge drinking, for example, as a target. It wasn't a target, but then some folks said, let's target binge drinking because that resembles some more of the animal behaviors that we treat than, relapse from abstinence. And so, because, right, what's relapse from abstinence in a rat? I'm not sure, but binge drinking, right, this repeated repetitive behavior. So maybe that's one of the reasons why there's been success in that. So. That's why I advise companies.
I think over the years there has been a bias against developing drugs for drug abuse disorders. Alcohol abuse, there has been quite a lot of basic research that suggests that you can develop alcohol antagonists that can prevent drunkenness and so forth. And there's been a real reluctance for industry to pursue those directions. Liability issues, what happens if somebody drives a car after they take a medication like this and gets into an accident? Would they sue the pharmaceutical company? Dealing with a drug addiction population, there are issues. So I think this is where we as academic researchers can really have a big impact. And I think the NIH recognizes this problem, and they've created certain initiatives to try to overcome this roadblock that we see in industry and have academics be more engaged and provide funding to support these. Maybe Chuck will talk about those.
move, drugs forward.
Yeah, because I think it's recognized that there's a huge societal impact and problem.
I mean, when I received my R1 that started in July, I received a call because they put the news from the lady. She said, my son is addicted to alcohol, he's doing medication, After when I received my R1, they put it in news and stuff for medication treatment. Then we received a call. A lady, she called. She said, "My son, he is sick and addiction to alcohol, and there is no medication." So I called her and I said, "This is too early. We hope in the future." But anyway, so
so psychiatry in biotech was completely dead. And then we had two rapid-acting depression treatments, esketamine, which I think is sort of was visionaries at J&J who had this little project. And I was wondering whether they were really serious about bringing it to the market or not. But they did. And it's groundbreaking. It's a incredible therapy with effect sizes in depression that we just haven't seen since the beginning of depression. And then brexanolone, Zulresso, again, huge effect sizes in postpartum depression, right? Again, and so it's kind of this companies going after things that actually just in drug abuse, intranasal naloxone. I mean, I met the pharmacist who got told by the firefighters and ambulance drivers that they were taking their naloxone and squirting it in people's noses to rescue them. And he said, this should be used. And he made a little company and got his nurse to be his sidekick on it. And when they actually showed it worked and turned it into a product, well, now it's sold. Right now it's commercial, but no company was going to touch that. That's why I always say, it took a champion. It took someone who saw the need, believed in it, and really wanted to see it go forward. And that's how so much of this gets done.
Yeah, and I think that just bears repeating, that this is where we as academics can fill the gaps. And that's the reason we have this course. And yeah, exactly. Okay, well, that was great, James. Thank you so much.